delta G and delta E are at the center of driving reactions. Enzyme regulation is at the center of control of metabolism in terms of whether a reaction is actually going to happen. So why do you have enzyme regulation? You need it to maintain constant levels of metabolites, such as ATP and NADH, in different cells. So if we look at metabolic pathway, for example, from PEG, Kyoto Encyclopedia of Genes and Genomes, we can see that there are many different pathways that are going on, and that they're related. So we've been dealing here, right, with carbohydrate metabolism, and then this is always good to orient for the TCA cycle, but you can see that there's a lot of connections. And in order to do all these things, right, to actually make energy, to make cofactors and vitamins, amino acids, lipids, etc., nucleotides, all these things have to be coordinately regulated. So in the cell, the major force that's driving everything is homeostasis. And for homeostasis, that means that things stay the same, right? So staying the same. So basically, one way to look at that is that intermediates are formed and consumed at the same rate. Yeah. So we'll look at that in a minute. So if you have a transient alteration, there has to be a way to compensate to return quickly to homeostasis. So this also relates experimentally. Often when we look at, we're trying to, especially like in my lab, where I look at perturbations, right, in response to pathogen or a stress, environmental stress, I'll often see big changes in transcription, but ultimately, if I measure all the metabolites in the cell, there won't be that much of a difference, unless it's making a very specialized metabolite, for example, as a defense compound. So typically, you'll see this one's have bigger changes in transcription in response to a perturbation than you'll see in terms of metabolite levels. That's not always true for these central pathways that we're talking about right now, like glycolysis, but it's true more in terms of like when you have a big shift or a big stress on the cell. Did you have a question back there? Where are you? Oh, sure. Is that better? Or the same? Better? I can't see who's asking. Now? Okay. So what I just said is homeostasis is what keeps the basically balance of metabolites, ATP and NADH the same in the cell. And so whenever you have a transient alteration, there needs to be usually a rapid way of getting back to homeostasis, particularly for central metabolism, what we're talking about right now. So there has to be compensatory changes to get back to that steady state. So typically, cells are containing like around 25,000 different proteins and thousands of reactions. So this is highly coupled and integrated, and as you'll see in this lecture, there's some key molecules that tend to regulate and keep things at homeostasis, and ATP is one of them. So usually the cell is always responding, right, because it's always changing, the conditions vary, and so there always has to be this way of continually keeping it at homeostasis, and a lot of this has to do with regulation. And in fact, of the number of genes in an organism, usually about 12% are involved in regulation. So when I was talking about this bottom part here about homeostasis, if you think about compound A being converted to S, being converted to the final product, right? You want to keep this flux constant, and you want to keep the concentration, for example, of S constant. And so you'll have ways of adjusting to keep this at basically running at steady state. And these are just velocities that you're seeing here. So for example, if something was perturbed where you didn't have enough substrate, then this velocity might be modulated to increase the amount of substrate to keep it at homeostasis. And we're gonna look at ways that you could actually change the enzyme activity and level. So enzyme activity can encompass the amount of the enzyme as well as the activity of the enzyme. People use it, always be careful. I'll be careful when I use it on tests and things, but when people are talking in general, they might say enzyme activity, and that can include both the level and the activity of the enzyme. Other professors, and it's again, still people in the field will only use enzyme activity when they're talking about how active the enzyme is, not expression. So just know that there's differences in how people talk about it. They might use it to mean both things or just one. So there are two things: metabolic regulation preserves homeostasis, and then metabolic control is a process that leads to a change in metabolic pathway in response to an environmental stress or a signal. So we're going to spend a lot of time actually on the next slide going through all of the different ways that you can control the activity of an enzyme. So as I mentioned, you can control the abundance, how much of the enzyme is there, right? That obviously dictates that, whether the ultimate activity of the enzyme, and you control the existing enzyme in terms of its activity. So we're all very familiar, right, with how you might control this. So here's our ultimate thing that we're looking at, the enzyme. So we have DNA being transcribed to mRNA. So I'm going to try asking questions, but who can tell me what regulates this transcription? Very generally, very basic terms. Yeah, you are right, but transcription factors is what I was looking for. So the reason I was looking for transcription factors is that transcription factors bind to specific cis-acting regulatory elements on the promoter, and those specific ones in front of your enzyme of, right, so you have the gene encoding your enzyme of interest, you have its promoter, it has, let's say, 4 cis-acting elements, and those transcription factors that bind to those cis-acting elements will control its expression. And you all had this in the first part of the course, correct? Yes? No? Seriously? Oh, okay. Okay, so I will back up. So basically, I'm going to have your GSIs make sure that you cover cis-acting, very basics of cis-acting regulatory elements and transcription factor binding. Just the basics of that's how you control it. So at that point, right, you have your messenger RNA that's produced, and it has to be translated to make enzyme. At this point, it can be turned over. This is a mode of regulation. And the degree of translation, the rapidity of translation can also be impacted. So for those of you who are in microbiology, right, this is different from bacterial protein synthesis where transcription and translation are highly coupled. In eukaryotes, you actually can have an uncoupling of these two processes. So here then we're at the enzyme. And the focus of this course, although the transcriptional regulation is also a driving force, we've been talking more about what happens in terms of the amount of the enzyme and its activity. So before I get to activity, whenever you have the amount of the enzyme, it's about how much is there, so that's transcription and translation, and then also its turnover. And a lot of times, actually, when we write papers, sometimes just to save space, people don't always talk about turnover. For example, if any of you have read papers on transcriptomics, so that's where we look at all of the mRNA in a cell, everything that's being expressed. And that we use things like RNA-seq, next generation sequencing, to do that. Sometimes people will just say, oh, the messenger RNA for that gene is upregulated. But actually, all you're measuring is the mRNA. So you don't know whether transcription was enhanced, leading to more mRNA, or whether the turnover was decreased, leading to more mRNA. So whenever you're looking at something, there's typically both something that's acting positively, like for example, transcription, and something that's actually altering translation and turnover, right, in terms of your messenger RNA. It's the same thing for the enzyme, right? You have these things coupled to synthesize the enzyme, but then we also have turnover. And turnover for different enzymes is actually quite different. So it can range from hours to days, and it depends on different tissue and the different function. And we also, in biology, have very specific ways of turning things over in a regulated way by ubiquitination and the proteasome. So these can be controlled as well. It's not just a process that's happening at a steady rate for every enzyme or for every even under every condition. So this was the ubiquitin proteasome, and then this is the lifetime from minutes to days. And again, in your book, they have a nice table of typically in different cells and different tissue, you might have different average lifetimes. And if you were ever interested, that is something that you can Google or look up what the average lifetime might be. Okay. So the other thing that you can have for an enzyme, so this is typically, I mean, it can be very fast if it's controlled, but it's typically a bit longer. This takes a bit longer, right? This typically takes at least 6 hours. It's more like, there are a few cases where it's very fast, but typically it's more like, to go from DNA to enzyme, it's more like hours to days. And typically it's more like 6 hours to, about 18 hours, because you actually have to transcribe the DNA and then translate it to get to the enzyme. And then we have some really fast ways of modulating activity. So basically, like I said, this one can be faster, but it's typically, if it's highly controlled, particularly by the specific ubiquitination. Faster ways that we typically think about, and these are involved, you're going to talk about a lot today in regulating central metabolism, are covalent modification of the enzyme. So this would be, for example, we'll look at it more in a minute, on a serine. So when you are adding a phosphate, right, so that's phosphorylation of an enzyme. So this is transferring like this high energy bond of the phosphate to the enzyme, right? You need an OH group. So there are 2 other amino acids that you can transfer that phosphate onto. Does anyone know offhand what they would be? So we have serine right there. Yeah, threonine, right? And one more? Tyrosine, okay? So you can only get phosphorylation on serine, threonine, and tyrosine. And so if any of you don't understand that, I do have a slide later, but then also you would want to go back, look at your structures on amino acids and review. And then different enzymes that do this covalent modification actually can have different specificities for serine, threonine, or tyrosine. So the next part of it that we're talking about is this fast control by allosteric regulation and substrate regulation. And we didn't put here too, but product regulation can also be an issue. So in this case, right, if we're talking about substrate, you can have a binding of a substrate. You can regulate it by having, so we're going to look at that in a minute. So the substrate regulation, it can be that you actually have cooperativity with the substrate. It can be just in terms of looking at the KM, the kinetics of the enzyme. You can also have competitive inhibition. If you have a competing substrate, that will influence how much of the particular product that you want. So there are multiple ways you regulate at the substrate level. Product inhibition, it tends to be, and you can even think of the thermodynamics of this, but typically if you have too much product, it will also limit the reaction. I'll show you on a reaction diagram. Allosteric regulation, I think you spoke about in the first part of class. Yes? Okay. So where's my, sorry, I'm missing my, there we go. Okay. So allosteric regulation is very, very important for central metabolism. It's very fast. You get, we call it an effector, EFF-ACTOR, that can bind to a specific site on the enzyme. It's usually not where the active site is, not where the substrate is binding, and it can change the conformation to make it more or less active. So this is very fast. It can be very fast, the allosteric regulation. The substrate regulation can be fast, and the covalent modification can be fast if the kinase that's there, that's putting on that phosphate group, is present. The other thing you can have is regulation by association with a regulatory protein. So you can have cases where a regulatory protein binds to your protein and either inactivates or activates it, or in some cases, it can also take it to another compartment, or a covalent modification or allosteric binding can take it to another compartment. In general, you just have to think of, I'll go over compartmentation, and you can think about these separately. We'll go through some examples. So for your eye clicker, I wanted you to tell me which would be typically the slowest regulation. And I'm gonna move this. I don't have it working yet.
For a quick transcription, right? Yeah, A. Wait, we should ask them the A.
Oh, sorry.
What? Yeah, she did in the, it's like the first one.
All right.
Debbie.
Okay, now it's going.
A, okay, cool. Oh, it's not on. Oh, it's not on. Okay. Alright, cool. I put A.
Do the A or B.
I already clicked A. I already clicked A. I don't know. I don't know. A? Hey, okay, yeah, it's A. Okay. Because he's going with B, and I'm like, crap. What?
Okay, so if you haven't voted, please vote now. I'm going to stop it. Okay, here we go. Okay, good job. So transcription is the slowest way, okay? Because you actually have to have the transcription factor, it has to bind, it actually has to transcribe, then you have to have the enzymes for translation, they have to be under the proper control, they have to make the enzyme, so that's typically a lot slower.
We all graduate.
One question.
Sorry. Okay. Here we go. So now we're going to take the basics of what we were learning. And there are a couple lectures where you're noticing I'm going slower. This is one because it relates. So the delta G was a slower lecture. And this part about regulation, I'm trying to go more slowly because it relates to so much of what we'll be talking about in the coming lectures. So we already talked about, this is why I switched the order of the lecture. We talked about glycolysis and gluconeogenesis last time, and now we're going to look at some ways that this is regulated. And you already know from last time, right, that there were 3 critical points, right? So there was this hexokinase entry, and again, that was ATP dependent. There was the phosphofructokinase entry, ATP dependent, or not entry, but regulatory enzyme, and then the pyruvate kinase. All of these actually were ATP dependent, and all of these had really high delta Gs, right? They were irreversible. So we can't have glycolysis and gluconeogenesis happening at the same time, right? Because that would be futile. You would be making something and then just converting it, and you're basically just losing heat. And even, we then looked at gluconeogenesis, and we looked at what we call these bypasses, right? How we bypass this enzyme. So we bypass it here, reversible, 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 reversible. We bypass here, reversible, we bypass here, right? And again, in this direction, these enzymes are irreversible, okay? The bypass reactions. So talking about futile cycles for one more thing, one, we want the whole pathway to be regulated separately. And then two, for each step, we want to make sure we're not just cycling and losing heat, right? So how do we do that? We have to regulate both simultaneously, and we want to avoid just basically expending ATP, right? And just generating heat. There can be conditions where this is useful, and bumblebees are one of them where they're actually using it to warm up in the morning. So they are doing a futile cycle. So, you know, in biology, there are always exceptions, and that's because there's a purpose, there's a functional purpose, and so that has been selected for. Okay. So what about how these specific reactions are regulated? So these lectures are interrelated and build on each other. So we're going back to this hexokinase. Really awkward findings. Yeah, here we are. Hexokinase right here. So remember the entry into glycolysis, glucose to glucose 6-phosphate. So this is really critical in terms of control, right? So we want to see how this would be regulated. So basically it's used for glycolysis in muscle for energy production, and in the liver for homeostasis. And in the muscle, you have negative regulation by the product. Okay? And in the liver, you actually have it being negatively regulated by a slightly different product, fructose 6-phosphate, which we'll look at in a minute. So whenever you see this, right, glucose 6-phosphate, sometimes I have just G6P, and this is fructose 6-phosphate, or it could be F6P. So in this case, it's called feedback inhibition or product inhibition. Okay? So you have your product, and that's inhibiting the enzyme activity. Here, this is a bit downstream of a product when you're looking at the liver, and it's inhibiting. So feedback inhibition can be the product, or it can be downstream in the reaction. And we see both of those things quite often in regulation of metabolism. Because sometimes it's like, okay, I've made too much of it, I don't need to move forward. And other times it's like, the whole point is to synthesize this product all the way like 10 steps down, and so that feeds back, okay? So if we look at a reaction diagram, well, what we're really looking at here is actually, pardon me, it's substrate, glucose concentration, versus enzyme activity. So you could think of this as velocity, right? So velocity here is Vmax, right, when you get saturating, or Vmax here. And then the Km is a half the Vmax, right? So that would be on this one about here, and then on this one, the Km would be here. So one thing that's really important for understanding biological systems and enzymes is typically they'll be operating around their Vmax. Now there are cases where this is a little bit different, for example, for hexokinase 1, but often if you know the Vmax, I think I might have said the wrong thing, typically they're operating around the Km, which is a half Vmax. So in this case, they would be operating here or about here. Okay. So the reason for that is that then they can be very responsive to the concentration in the cell. So when we as scientists are actually figuring out the kinetics of enzymes, we'll do these exact experiments, right, where we vary the concentration, and then we look at the velocity, and then we'll calculate a Vmax and a Km, and then we often will say that that relates to the substrate concentration physiologically in the cell. Okay. Here, this is very interesting because this is at the center of everything that we do in terms of regulation of glucose and glycolysis. So there are 4 different hexokinases. In the muscle, you have a number of them, and we're going to talk about hexokinase 1. And the enzymes have very different kinetics, even though they're the same enzyme. They have slight changes in their amino acid composition, which changes their structure slightly to allow them to have these different enzyme kinetics. So the hexokinase in muscle, you can see that at the 5 millimolar concentration here, it's already of the blood, it's already at Vmax. So it can actually be processing this amount pretty rapidly. Okay. Whereas in the liver, right, this amount that's in the blood is actually quite low. So we're going to look at the control. As you get flux going into the liver, it's going to be increasing its Vmax rapidly to be able to use that. Whereas here, you're already at this optimal point. We'll look at the next slide to go into that in more detail. So here's the liver. So basically what's happening here is a lot of this is also controlled by transporters, which are controlling the substrate concentration. So in metabolism, transporters also play a big role because they can control that substrate concentration. So here you have glucose in the blood, and then that's being transported by GLUT2, and usually its expression can be regulated. And then that goes into the liver, and here we're in the cytosol of the liver. It can be converted by hexokinase to glucose 6-phosphate and then to fructose 6-phosphate. So this has an interesting additional level of control, right? So it has the Km, we said in the liver, right? It has this Km. So we know how it relates to glucose in terms of control. But the other thing is that it can actually associate with a regulatory protein and be moved out of the cytosol and into the nucleus. So when you have fructose 6-phosphate accumulating in the liver, it actually will promote hexokinase moving into the nucleus with this regulatory enzyme. When you eat a meal, you have a lot of glucose, it gets transported into the liver. The glucose level is high, so that's what the positive arrow means. That's actually promoting dissociation of the regulatory enzyme with hexokinase to have it be on its own and to be in the cytosol. So you have this regulatory protein that's controlling its localization in the cytosol versus the nucleus, and the regulatory protein is dependent on that binding, the concentration of glucose versus fructose 6-phosphate. So the next enzyme in the pathway is the phosphofructose kinase 1.
Yeah.
Okay. So if we go back just quickly here, right, it's phosphofructose 1 here. So we saw, right, we had the glucose 6-phosphate, fructose 6-phosphate, and now we're here at this irreversible enzyme. So allosteric regulation is the primary method of regulation for phosphofructokinase 1. And later we're going to be talking about phosphofructokinase 2. So all through glycolysis we were talking about phosphofructokinase 1. So the 1 and 2 are important. So here, right, if you look at this, we look at the reaction and we're converting fructose 6-phosphate to fructose 1,6-bisphosphate. ATP is going in and then coming down, right? So basically, what this is doing for glycolysis, and we know in glycolysis that we're producing ATP, right? So if you have a lot of ATP, you don't need to do glycolysis because one of the major things it's doing is producing ATP. And so actually it acts as a negative regulator, and it's an allosteric negative regulator. Whereas low levels of ADP and AMP mean I need ATP, right? My energy reserves in ATP are low, and so I need to do glycolysis. So again, you want to think about the reaction of glycolysis, that net production of 2 ATP, and that if you need ATP, then you would have low ADP and AMP, and that would be positively regulating flux through this pathway. And it's positively regulating it by binding to the enzyme at an allosteric site. Whereas if you have a lot of ATP, it could be a little counterintuitive because you might think, oh yeah, I have ATP, I can use it. But this is about regulation of the enzyme, and you don't want to go through all of these enzymatic pathways if you don't need ATPs. Yeah. Okay, when I use the word flux, and when other biochemists use the word flux, we're meaning the rate that basically substrates to products and substrates to products is moving through. Okay. Your book, I actually love that area. It's called like metabolic control theory. Your book has a section on it, but I didn't assign it because it's not as critical to this level of biochemistry. For those of you who are chemical engineers or who get really into modeling in biological systems as well, you do want to get to know metabolic control and flux. And it's specifically using the word metabolic control in a specific way. Okay, so the regulation of phosphofructokinase by allosteric. So usually when you have an allosteric interaction, allosteric regulation, you have an enzyme that has either multiple subunits of the same thing, so it could be like a homodimer, same dimer 2 of the MER, the protein subunit. So homodimer would be 2 of the same, or you could have a heterodimer where you have different subunits coming together. So in this case, right, for this enzyme, we actually have 4 subunits, 1, 2, 3, and 4. And they each have a substrate site. So in this case, they're similar, and they have the same substrate site for ADP and the fructose 1,6-bisphosphate. And then they also have this binding site, ATP. And so what can happen is that actually it's changing the kinetics of the reaction. So according to the amount of fructose 6-bisphosphate with ATP high versus low, right, when you have high ATP, you have this slower, this more spread out reaction, right? A lot slower, and you have to get to a lot higher fructose 6-phosphate to have a significant activity. Whereas here, right, with low ATP, it's very rapid. So if we look back at the reaction, right, fructose 6-phosphate going to fructose 1,6-bisphosphate driving glycolysis, right? If we have the same amount of fructose 6-phosphate in the cell under low ATP, so let's say we're here, you would actually be making your product, right? So this is the velocity or the activity of the enzyme, and so you would be making a lot of it. And here you're actually even saturating it. But if you had high ATP, you'd be basically doing nothing because it doesn't want to waste making the next product, going on through the reactions. And it's really, you want to be controlling it, right, at this irreversible step. Otherwise, if it goes on, these things are at equilibrium, it could keep moving forward. So you really want to control it here, and that's what it's doing. So the next one that we're going to look at is here, where are you? Pyruvate kinase. Oh, actually, sorry. There's something in between. Sorry, going back here. So we talked about phosphofructokinase, but but we didn't talk about its parallel reaction, the fructose 1,6-bisphosphate. And these are coordinated. So if we look back here, right, so if we're going this way, right, to fructose 1,6-bisphosphate, we're going down to pyruvate plus ATP, and if we're going up this way, we're actually making glucose, so gluconeogenesis. So how do we control what happens at the point of this fructose 6-phosphate? So it's actually coordinating gluconeogenesis, making glucose, and glycolysis. So as we showed here, right, this is the irreversible reaction glycolysis, and then this is the bypass for gluconeogenesis. Here you're using ATP, but if you have high ATP, it's strongly inhibited. And that's because you don't need the ATP made by glycolysis. If you have ADP and AMP, it's favored because those are in those they store less energy. They mean that you're, in terms of the energy metabolism of the cell, that you actually need to make ATP. So you have a parallel reaction, coordinated regulation of gluconeogenesis here. So this is the fructose bisphosphatase. So in this case, AMP inhibits this reaction. So when ATP is high, this doesn't happen well. When AMP is high, this doesn't happen well. So it's a way of coordinating what's going on.
I don't think it affects.
I have a question.
Yeah.
Do ATP and ADP have an effect on the glucogenesis?
I'm sorry?
Do the concentrations of ATP and ADP have an effect on the glucogenesis reaction?
Do the ATP and ADP? Not on this specific enzyme.
Okay.
So when we're talking about this, there's another element to this, which is actually, in addition to ATP and AMP, there's another allosteric effector, and it's called fructose 2,6-bisphosphate, right? And you guys all know how to name this now, where you have the fructose, and then the bis means the phosphates are different directions, or different positions, and then you have one at position 2 and one at position 6. So this is not an intermediate in the reaction. I know it looks and sounds like other things, but it actually, it is not. And so sometimes with allosteric effectors, you'll have another product that's regulating. So we have ATP and AMP, but then we also have fructose 2,6-bisphosphate. And basically, it's reflecting the glucose level in the blood, which we can look at in a minute, positively regulating one, negatively regulating the other. So not only do you have the ATP, AMP, but you also have the fructose 2,6-bisphosphate. So if we look at this, right, we can see what's going on in the two enzymes from the allosteric regulation. So for glycolysis, right, the affinity is increased. Again, if you're at, so now we're at the substrate concentration, fructose 6-phosphate, and when we have this allosteric effector, wow, we can really do a lot, right? And usually, again, this is a conformational change that's influencing the enzymatic rate of the enzyme. In the case without it, right, this is very minimal in terms of what's happening. I'll stop after this one here, and I'll go back over this again next lecture. So for the opposite one from gluconeogenesis, right, you have the opposite thing happening. Very low with the fructose 2,6 and very high without it. So, for example, on an exam, if I gave you that this was a central regulator of these two pathways, you might need to be able to draw these two different diagrams. And we'll discuss this again because some people have to run. We'll start on this slide for next lecture.